My name's Bill, and today I'm going to be talking to you about obtaining access to controlled data in the GDC. I'm the lead for the GDC User Services team, and we get many questions through our help desk about controlled access data all the time and how to retrieve this access. So I hope this will be useful to all of you users out there. To start with an agenda, we're first going to give just a small introduction to GDC data. I'm sure lots of you know this stuff already. Then we're going to talk about obtaining controlled access, how that whole process works, and how to use the controlled access data once you do get the correct authorization. Following that, we're going to talk about a few controlled access data sets that are present in the GDC, and then we're going to go on to some frequently asked questions. So starting with data at the GDC Data Commons, as you probably know, the GDC is a data repository for cancer genomics. This includes a few different types of data, mostly molecular, biospecimen, and clinical. In terms of access, GDC data is divided into two categories, open and controlled access data. The thing that I wanted to specify when I say it's divided into two categories is that controlled access is on a project level. So if you have access to a certain project, you have access to that. But if you need access to two separate projects, you will need two separate access levels. The open data in the GDC, there is no login or special access that is necessary. Anyone can download the data. You don't need to log in. You can go right onto the GDC website, click download, and get whatever data that's open you would like. This usually corresponds to data that is unidentifiable or derived so that no actual genomic sequence is present. And typically it's so that subjects can't be, reverse engineered and identified based on this information. Controlled data requires special permissions through DBGAP. I'm going to start talking about DBGAP in a little bit, but this is typically identifiable data. A lot of these do contain molecular sequence or things that can be used to infer molecular sequence. And as you know, a lot of times when you know somebody's specific sequence, you can trace it back to a specific person. So that's why this data is controlled. The purpose of having this data controlled access is to keep data that's from human donors available to those who need it while preventing the data from being accessed carelessly or maliciously. The GDC adheres to the NIH Genomic Data Sharing Policy and also the NCI Genomic Data Sharing Policy. All authorization to controlled access data is received by the GDC through the NCBI database of genotypes and phenotypes, and henceforth I will be referring to that as DBGAP. One thing I want to stress here is that this process is automated. The GDC will get a report telling us who has access to which projects. That is fed into our system through our login. So when you log in, you will automatically see which projects you have access to. If that doesn't match up to what you think it is, we must get the automatic system working before you will be allowed to download any data. We don't make any exceptions, even if you do have some sort of proof that you should be able to download the data. So if you do find yourself troubleshooting with us, just want to let you know that. So jumping right into obtaining controlled access, I want to start out with a few caveats because the first steps to this process are not actually through GDC. They're from separate organizations. That includes ERA Commons and DBGAP specifically. They might change their interfaces and their procedures and anything from their end. So we're not going to go into too much specifics about that here because we are GDC affiliated and don't really control this. But we recommend contacting their help desk if you have any specific questions about their procedures. If you're ever in doubt and you're looking to get access to GDC data, you feel free to contact the GDC help desk, and we can usually point you in the right direction for which help desk you should be contacting. So we might not be able to help you directly, but we can definitely point you in the right direction. So if you go on the GDC website, you may have seen this diagram before. This is adapted from a similar one. These are the basic steps for how to obtain controlled access. So first, you must obtain an ERA Commons account. Using that ERA Commons account, you would obtain DBGAP access. You would then obtain access to research projects through DBGAP as a PI. And then as a PI, you would grant access to individuals in your lab so that their ERA Commons IDs are associated with your project. And then finally, when you log into the GDC Data Portal, these access levels should be there. So getting into the specifics for that diagram, starting with ERA Commons, this is your username. This is what you use to log into the GDC. There's not a separate GDC login. You will log in through the NIH interface. This is required to apply for data access through DBGAP. And this is what you log into both. This is what you will use to log into both DBGAP and GDC. You might hear it called different things like your NIH login or a DBGAP login. They can be the same thing. There's a possibility that some people have more than one. And if you have access to more than one project and perhaps you're logging into GDC and not seeing what you expect, I recommend contacting the PI of your project because they know exactly which username is associated with the access that you should have. So now moving on to DBGAP, we're going to talk a lot about DBGAP, but just to give some basic information about how it relates to GDC, DBGAP holds information on which researchers are authorized to download each dataset. So like I said, DBGAP sends a report to GDC and it says, this person has access to this project, and this project, and not this project. Well, the GDC automatically retrieves this, and this is what you will see when you log into the GDC. So when you want to apply to DBGAP to access a dataset, first you obtain access to DBGAP. This is automatic because once you have an ERA Commons ID, you should be able to log into DBGAP. Then the PI of your research project will create a research project in DBGAP. So this step is mostly to provide information to DBGAP to determine your project's fit with each dataset. Each dataset will have a list of rules and specific guidelines that must be followed. And when you provide information about your research project, the DBGAP officials will take a look and make sure that your project isn't looking to do something that the dataset doesn't authorize, or to make sure that these subjects match in general. Once you do have a research project in DBGAP, you'll start applying to access for each dataset. So you will have your research project, but you can apply that research project for access to multiple datasets. And one question you might have is, how do I know which dataset in the GDC I should apply to? And the answer is that you would use the DBGAP accession, and that's also called a PHS ID because it starts with PHS. So I'm going to show a really quick, not quite a demo, but let me see if I can get out of this. Just to show you how to know which PHS ID to apply for if you're logging into the GDC. So this is the GDC Data Portal, portal.gdc.cancer.gov. And what you would do is go to the projects page. It can be done here or at the top. You would perhaps search for a specific project using these filters. In this case, we'll look at one of the TCGA projects. I do know the TCGA projects are kind of all within this one PHS ID, but every project will have them listed. So here, if I want access to TCGA Brica, I would know that I should gain access to this DBGAP accession or PHS ID in DBGAP. So every project in the GDC has a page like this, and you can figure out which ID you need with that. You could get this information from the API too. So if you need like all of them, you could do that. But this is a pretty easy way to figure that out quickly. So going back to the presentation here. After you apply, the status of each application can be found under the My Request tab. So that'll tell you which step you're at on the DBGAP's end, whether somebody's reviewing it, whether they just received it, whether you need to press another checkbox to make sure things are going through. And then once the application is approved, lab members can be added to the project by the PI. So the PI will have access, but the lab members will get access to the project's dataset through their own ERA Commons ID. So one thing that I've seen before is usually postdocs, grad students. I think those are usually pretty okay, but sometimes there's a lab member who doesn't quite have a usual relationship, like a visiting scholar or something. And sometimes we get questions about that, and GDC can't really answer those. So if you have any questions, I recommend contacting the NCI Office of Data Sharing, and their email is right down here. And I do want to add, by applying to gain access to a specific data set, you agree to abide by the DBGAP project's data use agreements. So they will all have those, and you'll be able to review them as you go through the application project process. So one last thing, I'm not going to get too deep into this, but any submission access to GDC requires controlled access. You need to gain access to submission. There's no open access GDC submission. So if you are a GDC data submitter, your group is going to be submitting something to GDC, you will need special access to do that. This cannot be given directly by the GDC, but it needs to be provided by a project's DBGAP Genomic Program Administrator, or GPA. This is somebody who is in the DBGAP database for each of those DBGAP accessions, and each of those each data set has a person who can give you this access. So I would recommend contacting DBGAP if you don't know who that person is. So next, I'm going to talk about how to use controlled data in the GDC, and how to actually access things that are controlled. Everything I'm going to go through here is going to be, I'm going to show you on the presentation, but then I'm going to do a quick demo afterwards. So if you have any questions about that, hopefully the demo will answer it, but also happy to answer afterwards. So before I talk about the specific ways to download, I want to introduce the concept of the GDC token. The GDC token is just an alphanumeric string that can be used to authorize your access. So in order to download one of these, you will click on Login at the top right. You'll use your credentials to log in to the NIH pop-up that comes up. You will then hover over your username. Sometimes you have to click it, depending on which browser you're using. And you will just click on Download Token. And then a TXT file will download to your computer, and the token string will be inside that TXT file. So another question you may have when you do have controlled access is, which projects do I have access to? You'll probably know this ahead of time, but this will tell you in the eyes of the GDC what you have access to. So once you log in, you can click on User Profile, which is right above Download Token, and you will see a table there with a bunch of columns and some check marks. The only one that you should be concerned with is the one that is underscore member underscore. If you have a check mark under this, you have authorization to download controlled access data from that project. So look for that. I also want to talk about GDC Token Safety because these are important to gain authorized access to GDC controlled data. You want to be safe and you don't want to do things like share your token with anyone else or with the GDC help desk. Do not send us your token. There's something I do want to tell you, and that's that downloading a new GDC token will invalidate the old one. And I want to tell you this for two reasons. One, if something happens and you feel like your token might be compromised, always follow what your institution tells you to do in terms of security. But a good first step is to go and download a new GDC token right away. That'll invalidate any old one, and your new one will be the only one that works. The other reason I want to tell you that is because if you are running a long process that requires controlled access, like a long upload, download, or you have a pipeline that downloads from GDC, if you download a new token, the one you're using on your pipeline will be invalidated, and you'll just find a bunch of error message when you come back after the weekend. So I just do want to warn you about that. It's happened to me 100 times, and I would not like it to happen to you. Lastly, I would like to let you know that tokens expire 30 days after downloading them. So if you feel like your token might be getting stale, go ahead and download a new one. Not a big deal. So now you might be asking, where can controlled access be used in the GDC? I told you how to get the token and what to do with it, but now you might be interested in downloading data. Of course you are. So we have three main places where controlled access can be used. We have the GDC Data Portal, the GDC Data Transfer Tool, and the GDC API. So with the GDC Data Portal, you don't actually have to download a token. When you log in, your information, so you won't actually be handling a token. The information for what you're authorized to download is stored in your browser session until you are logged out. So if you log out, you'll have to log back in. If you go into like an incognito window or something, that will also not contain your controlled data. So if you want to be extra secure, only log on with an incognito window and then close it afterwards, especially if you're using perhaps a public computer. And make sure all incognito windows are closed. Those can move across sessions. If you've watched previous webinars, you might have seen this slide before, but this is just a basic outline for how to download controlled data for the GDC Data Transfer Tool. This is specifically for the command line interface. The GDC token can be passed to the GDC Data Transfer Tool as a file. So you would just run this command here, GDC client download -m manifest.txt, which is your presumably contains some controlled access data, and -t and then the token file. You don't have to put the token string here or anything. You can just put the file in which the token is that the token string is inside. So you would do this, and if you're not downloading any controlled access files, this whole part is completely optional. So the Data Transfer Tool UI, it's basically the same thing except it's with a UI. So you'll see on the top right, if you don't have the token file ready yet, it'll say no token file. You will click there and you will have this nice little interface comes up. And if you can now drag the token into this space, or you can click the plus button and browse to it. And then if you plan on downloading a bunch of stuff soon after, you can click to remember the token after the program is restarted, and saving the token is how you can then start other downloads. So lastly, I want to talk about the GDC API. if you are downloading something, a controlled file through the API, you need to pass the token to the GDC API as a variable in the header named XAuth token. All the examples I'm going to show you are in curl, but there are plenty of other ways to do this. So one thing we do is we recommend assigning the token string to the variable without copying and pasting the string itself. So if you copy and paste the string itself, like if you do export token equals and then you copy and paste, that'll work, but your token string will be in your history, which is less secure. So we recommend using this $ parentheses and then cat, which prints to which will print your token string, and then put this directly into this token variable. So it won't show up in your history. So then when you run a curl command, you would just use -h for header. XAuth token is your key, and then your value would be the token variable with the $ in front. And that should allow you to download controlled data from the API. Oops, that's not the button to go to the next slide. Okay. So real quick, I want to walk you through all of those steps. So in terms of downloading a GDC token from the portal, very basic if you've used the portal before, but you would just go ahead and log in. An NIH login will come up. I have my creds saved in here. And once you log in, you don't need to be on any of these like repository or projects. You would just click on your name and you can click download token. I might have some things running right now, so I'm not going to click download token right now. So the other thing I want to show you is now that you're logged in, you can add some controlled files. In this case, I know I have access to these. So I have 2 controlled and 2 open files in my cart right now. I will go to the cart, and all 4 are authorized because I do have access to all 4. Now, if I were to log out, you will see that this changes to 2 authorized and 2 unauthorized because now I've lost my token information, and now I'm just anyone from the public. So I'm unauthorized to download these 2 tokens. So going along the presentation again, we have which projects you have access to. You would see that simply by going to I'll log in again. And just clicking user profile. So I've showed this. So if you're using the data transfer tool, I'll bring up the terminal real quick. You would just run, I'll put it down a little bit here. GDC client download -m manifest.txt. It's an imaginary manifest. And then -t, you would put your token file. In this case, you would put here, I have this token here. It's called GDC Token Fake because it's not actually a token. Now, if you want to see what the token looks like, I can show you. This is the name of the file, GDC token, and then I put a fake token in here as I wrote here. This is not a real token because I would not want to expose that to this public webinar. You'll just open and you'll see a bunch of alphanumeric characters. This was just me randomly typing a bunch of stuff and copying and pasting things. This has nothing to do with the actual structure of the file. So if you wanted to put a token into the API, let's say I wanted to put it into a variable called token var for variable. So I have nothing in there right now. I can check by using the echo command, and there's nothing in there. So now I want to put what was inside this file into that token variable. So I would do the export command, token var equals, and $ parentheses, cat, and then this file right here. I'll just copy that in here, close parentheses. And as you can see, there is no output. It doesn't show the actual token, but if you were to look into the token variable now, you will see that it is in there. So now you are ready to do an API call. And in that case, you would start out your curl with the header command. You would use the x auth token key, and then token var contains your token string. So the rest of your API call would come after this. If you have any, I'm not going to actually run it because it's not going to work. But if you have any, if you're wondering how to actually perform these downloads using the DTT and API, we have plenty of past webinars that will show that, and this will be recorded too. So lots of webinars too that are recorded that you can check out. So going back to the presentation here, I want to talk about some controlled access data sets that are in the GDC. So we have right here, I just listed several types of files that the main types of files that are controlled access in the GDC as of this date, January 31st, 2022. This includes all BEM files in the active portal, all variant calling files except for the masked semantic mutation files for certain projects. These are authorized we're authorized to display variants from certain projects that give us permission and have gone through a special pipeline that removes a lot of the potential germline variants. And then we also have the splice junction quantification files from RNA-seq that are controlled access. There are others that are kind of in smaller groups, and you can check out what those are by going to the portal, going to the Files tab on the left, and just going down to this Access facet. And you can click on just Controlled or Open if you're looking for that, and this will just show you all of the different file types that are controlled access and for which projects. So if you're wondering, that's a good way to figure it out. Next, I want to go through a few of the kind of interesting data sets that I want to highlight that are controlled access in the GDC. And if you watched one of our webinars it was more than a year ago, it was on GDC data, so you might recognize some of this information from there. So Foundation Medicine, we do have data from them. They're a molecular information company that specializes in precision medicine. They generate genomic profiles for thousands of cancer patients designed to match each patient to a personalized treatment plan. That's kind of their whole goal. So FMI, they provided the GDC with genomic profiling data from about 18,000 adult patients with a diverse array of cancers that underwent genomic profiling using their commercially available Foundation 1 genomic profiling assay. So yeah, we have data from this particular assay. And just to give you a little more information about what is in the GDC, this Foundation Medicine dataset is named FM-AD for Foundation Medicine Adult Dataset. Here's the DBGAP ID. We have 18,004 cases across 42 primary sites. And because this is an assay or a panel, this is classified as targeted sequencing in the GDC. And this data is available as raw VCF format, annotated VCF format, and MAF format. The second one I want to highlight is the AACR Project Genie. The GDC provides about 45,000 cancer patients data that corresponds to the AACR Project Genie version 5.0. The Genie projects are broken up into 8 contributing sites. So when you see them in the GDC, you'll see Genie as a program, and each of the 8 contributing sites are projects. They have clinical biospecimen, mass somatic mutations, copy number scores, and transcript fusion data available. The clinical data from Genie was mapped to the GDC data dictionary, and the genomic data that Genie provided was mapped from the HG19 genome to the GDC Reference Genome HG38. You may have heard the GDC uses that reference genome specifically. And just some specific background about that in the GDC. Here are all the projects. As you can see, you might recognize some of these as institutions that do cancer research. There's 44,756 cases across 49 primary sites. And this is also a panel based data. It's recognized as targeted sequencing. It's classified as targeted sequencing in the GDC, and it's broken up into MAF files, which are the simple nucleotide variation data, and then TSV files for the copy number variation and the structural variant data. So now I'm going to get into some frequently asked questions that the GDC help desk receives quite often. So one question that we get a lot is, can researchers who are not based in the United States obtain access to the GDC? And I put yes here because that's the answer, and if anyone misses this question being answered, I want them to see it. Researchers who are not based in the United States can obtain access to GDC controlled data. I think there's a few extra steps to get an ERA Commons account. If you are in that category, I would suggest reaching out to them. But I think in general, it's mostly the same process as researchers who are based in the United States. Yeah, an ERA Commons would be able to straighten out exactly what needs to be done if you are not in the United States. So when I try to log in, my username and password disappear with no error message. So sometimes this happens. You type in your username, you type in your password, you click login, and it just takes you right back to the same page you're on, but your username and password are now gone. This is alarming sometimes, but this is actually a quirk of the login system. This usually signifies that your ERA Commons account needs to be the password needs to be reset. So to fix that, I would go to the ERA Commons website, click on the forgot/reset password, and then your username and password should be working once you go through that process. Okay, so the next one, when I try to add my lab members to a DBGAP project, they do not show up in the interface. And that's a very simple solution. So when you're adding people to a DBGAP project, you need to type their name into a field, and then their name will pop up so that you can give them different permissions. Sometimes their name won't pop up, and this is usually because the lab members need to log in to DBGAP one time before they can be found in this interface. So if your lab members aren't showing up, just make sure you're using their full name and ask them to log into DBGAP. That should fix the problem. So the last question, how can I download GDC data from DBGAP? So one thing, if you're familiar with DBGAP, you may have noticed that most of what I've been talking about refers to getting access from DBGAP and not downloading data from DBGAP. And I know for many projects, data is downloaded directly from DBGAP. And that is actually not the case for the GDC. All of the projects that are hosted in GDC their data can be downloaded from GDC and not DBGAP. I know that some projects do have some supplementary data that may be in DBGAP, but most of the molecular data will need to be downloaded from the GDC API or portal, whatever you're using. So they can't be downloaded directly from DBGAP. So lastly, I want to show you a troubleshooting flowchart. So this is the basic flowchart that my team who runs the GDC help desk uses. So you're a user and you get an error message that suggests your token is not valid. So it could be this one. It could be saying one that your token isn't present, or yeah, there's a few token-based error messages that you can get. But troubleshooting them usually follows just the basic flowchart. Your first step, try downloading a new token if you can and see if that works. Sometimes that does the trick, other times it doesn't. So the next step is to check access on the portal. And when I say that, what I mean is, like I showed you before, I would go to the portal, go to my username up here, and click on user profile. Whatever you have access to in your user profile is what you have access to in the eyes of the GDC. So you will want to check that out, and specifically for the project you're trying to download from. So once you check that, if you have access, then I would recommend contacting the GDC help desk. This means that for some reason, your access isn't acting correctly, and this could be a problem with a command, or it could be something else, and it is likely on the GDC's end. So we're happy to help with that. Now, if you go on the portal, if you check your access on the portal, and you don't see, you look at your account and it says you don't have any access to DBGAP projects, or it says you don't have access to the specific projects you thought you did. In that case, there's not much that GDC can actually do to help, because all of the access needs to be gained directly through DBGAP. So for that, if you have a PI, I would contact them to see to make sure that they added you correctly to the DBGAP project. And then if you are the PI and you do believe you should have access, I would contact DBGAP, and they should be able to sort out why you're not showing up in the reports that are coming into the GDC. So the last thing I want to show you is a few GDC links. And before we do that, Sharon, I wanted to ask, how many questions do we have?
I don't think at this point, Bill, that there are any questions. Oh wait, I'm sorry. There was one in the chat. I'm looking at the Q&A. Do you host the ICGC controlled data?
So my understanding is that there's a little bit of overlap between some of the projects that are hosted by ICGC and GDC, but we don't host anything specifically from ICGC. So there could be some overlap in data sets. I don't know for sure. I'm not too familiar, but we don't specifically host that.
Thanks, Bill. And I'm sorry, there is one in the Q&A section. There's a question. If I want to download data for RNA-seq that are aligned and pre-processed, i.e. the level 3 data, will that be individual data for each sample, or is a collated data available for download?
So right now we have things available as individual samples because we allow users to download specific subsets by kind of on an aliquot level. So we couldn't possibly have every combination of collated data in there, so it is not collated at the time. Eventually down the line, we will have the data collated, but if you were looking to download right now, we do have a tool called the GDC RNA-seq tool in our community tools page, and that can be used to generate a matrix of RNA-seq expression values using a manifest. Though I will warn you with an upcoming release that may not function the same way. So yeah, I think that's it.
Okay, Bill, a couple other questions came in on the chat. The first is, do you need to be a PI to set the account?
So I believe you need to be a PI to apply for specific data to apply for specific like data sets in a research project. You do need to have the PI designation within DBGAP. Now, whether or not that specifically corresponds to having a PI designation in like a university or your institution, I don't know the answer to that. So I would recommend contacting DBGAP for that. But most of the process in DBGAP must be done by a PI, at least in DBGAP size.
Okay. I think there's one other question, but it's related. Is it possible for a PDR to set the account? And I think you've just answered that with the PI.
Okay, cool.
So are there any other questions?
All right. So one thing I do want to show you, because this was produced by DBGAP. This is a sorry. This is a video produced by DBGAP. It's less than 5 minutes long, and it describes the process for applying very well. So I am going to bring that up. And if you don't have time and you need to leave, you would get to it by going to access data, obtaining access to controlled data on the left side here, and then scrolling down just a little bit, and you'll see this video here. So I'm going to actually click on it to go to YouTube so it can be bigger, and I will full screen this, and we will watch this for 4 minutes.
Tutorial walks you through the steps to request access to control data in DBGAP. Overall, there are 4 steps. The first, which is covered in this video, is your application for access. The second step is the approval process. The third is downloading the data. And the fourth requires that you either close out the project or renew it annually. The closeout and renew steps each have their own video tutorials. I'll start on the DBGAP homepage by clicking this link, Controlled Access Data. Here you can learn how to set up an account at NIH's ERA Commons, or go directly to the login page. I've logged in, and we are now looking at my mock project request, and I'll go ahead and click Create New Research Project. It's a good idea to read the section before you get started to be sure that you have the information you will need, and of course read the User Code of Conduct. When you're ready, click Begin New Research Project. You will step through these tabs, where the darkened tab is the page you are currently on, and you can access pages where the tab label is blue. By the way, this link to the Office of Management and Budget control number refers to the public reporting burden for this process. Since I started a new project, I have to first choose the datasets that I want to access. Study names are roughly alphabetical. I'll scroll down and check this IBD only study. Note that this consent group requires Institutional Review Board or IRB approval. We'll see later on the Review Data Use Certificate page how to attach that approval document. I'll also select this ARIDS dataset, then scroll down to the bottom and click Add Selected and Continue. Here you will input your project details. On all of these pages, fields with an asterisk must be completed, and the blue question mark links offer good advice on what to include in a field and also how that information will be used by DBGAP. I'll complete the required fields, select a signing official. I'll move to the next step by clicking Save and Continue. If you have no collaborators, you can skip this step. If you are adding external collaborators, that is, those from other institutions, be aware that they must submit a separate data access request, and uncheck the box for none to fill in the required fields. I'm not adding any here, so I'll go up and click the IT Director tab. Notice that you now have access to the last two tabs. You can use these pages at any time. For example, the Data Security page allows you to reset your decryption password. At least one IT Director is required, and basically this is someone responsible for the computer systems where the data sets are to be stored. I've added my director, so I'll click Save and Continue. For this video, I started a new project and selected my data sets in the first step, so I have skipped to Confirm Datasets. I'm satisfied with these consent groups, so I'll just click Continue. And we stress that you carefully review the data use certificates at this point. This is where you would upload any required documents. In my example, that would be the IRB approval form for the NIDDK study. Notice that the files must be in PDF format. I'll click Continue. I must now affirm that I've read and agree with the terms of the data use limitations for my datasets. So I'll check these boxes and click the I agree button. We're now on the last step. Here you should review your application and either approve or revise it. I'll check these boxes to approve and submit my application to the signing official. You will receive emails at each step of the approval process, and you can come back at any time to check the status on the My Requests page.
Oh, okay. Sorry, I'm trying to click on the... Here we go. Okay, well, that's the end of the presentation. Thank you all for joining. And here are some GDC links that many people find useful. The top one is for GDC documentation, which are the user's guides, then followed by the GDC website, which has higher level information. The video which I just showed you is on that website, or it's linked to on the website. It's not actually hosted there. The GDC Data Portal is portal.gdc.cancer.gov. You can find some more information about controlled data access at that page I just showed you before, including data access policies. And if you want to, in the future, watch the recording for this webinar, or now watch the recording for previous webinars, you can go to the GDC webinar page on our website, and you will see their videos there. Thank you all so much. Are there any other questions?
I think, Bill, you just addressed the question about will slides be shared with everybody, and the video itself is going to be posted on the webinar page once we finish processing. Okay, any other questions? Okay, I think that's it. Thanks so much, everybody, for attending. You all have a great rest of the day.
Thank you, everyone. Bye.